Hi, my name is Joseph DeRisi, and I'm an associate professor in the Department of Biochemistry and Biophysics and a Howard Hughes investigator at the University of California, San Francisco. The subject of my lecture today is malaria, and it's my goal to give you a background and general overview of the disease and the parasites that cause this deadly affliction. So first, let's talk about the disease. The disease malaria is a devastating condition. It's caused by one of four parasitic protozoa. It's characterized by a periodic fever or a febrile incident. This is usually followed by chills, sweats, aches, and other devastating conditions. It is also characterized by enlargement of the spleen, and in the worst form, so-called cerebral malaria, can result in seizures, coma, severe anemia, and ultimately organ failure and death. It is true that children are the most affected by this disease and most likely to die. The toll of human malaria is incredible. There's somewhere between 1 and 2 million deaths due to this disease per year. And that is in addition to 400 to 900 million disease incidents per year. So let me put that in perspective for you. 1 million deaths per year equates to someone dying of this disease every 30 seconds of every minute, every day of the year. And so it cannot be understated how incredibly brutal this disease is on humanity. So why is malaria such a problem? Why are we still dealing with this today when the cause of the disease has been known for over 100 years? Well, it's simple. Today, malaria enjoys worldwide drug resistance. That is, the limited medications that we have to treat the disease are useless in many parts of the world, and I'll have more to say about this in a little bit. There is currently no vaccine for this disease with any operational impact. And to acquire some limited immunity to the disease requires that people be repeatedly infected on a very frequent basis. Therefore, to have any immunity to the disease requires continual pressure by the parasite. No vaccine that has been created today achieves the same level of protection as natural infection. And that's a problem. So this is where in the world the risk of transmission of malaria is the most severe. Colored in dark green are those areas of the most intense transmission of the disease. And as you can see, here in sub-Saharan Africa, the problem is the worst. Now, this is not to say that the problem isn't bad in South America, Central America, and Southeast Asia. It is, but by far and away, the largest burden to malaria is in sub-Saharan Africa. And the disease malaria is transmitted by the Anopheles mosquito. There are over 60 species of Anopheles mosquito that are capable of transmitting the disease. And this is why it is so difficult to control. You might think that one way to control the disease would simply be to eradicate the mosquitoes that carry it. This is what has worked in many first world countries that have the organization, the money, and the governmental structure to support such an effort. And in fact, during the 1950s to the 1970s, there was a worldwide global eradication effort led by the WHO, the World Health Organization. And here's a postcard from the United States commemorating the initiation of the worldwide eradication effort. But since I'm giving you this lecture, you know that this eradication effort was doomed to failure. And why did it fail? It failed for a variety of different reasons that are illustrated here. First of all, as I mentioned before, there's drug resistance to the medications we use to treat the sick. Furthermore, a lot of the eradication effort bypassed Africa, where the worst problem of malaria actually was occurring. Furthermore, there was a lack of sustainable funding for this effort. And along with that, a lack of sustainable community participation in the eradication effort. This was compounded and complicated by the fact that there was massive population movements throughout the world in areas endemic with malaria and war, which never helps anything. Furthermore, insects became resistant to many of the insecticides used to try to eradicate the vectors themselves. So what about the parasites themselves? What is it we're trying to treat? Well, as I mentioned before, the disease malaria is caused by any one of four protozoal parasites, and their names are here. The worst, and far the most deadly one, is Plasmodium falciparum, followed by Plasmodium vivax, Plasmodium malaria, and Plasmodium ovale. Plasmodium falciparum, as I mentioned, is the most deadly of the four and has the largest distribution worldwide. Now, if you were to take some blood from an infected individual, smear it on a glass slide, and stain with a dye that binds DNA, you would see something like this. Now, as you may know, red blood cells, or erythrocytes, do not contain nuclei, and thus do not contain DNA, but the parasites do. So, if we did stain with a dye that bound DNA, you would see healthy red blood cells, such as this one and this one, but you would also see red blood cells with dark objects on the inside, such as here and here. These are plasmodial parasites. In fact, this is Plasmodium falciparum. And this is the most frequent way an infection by Plasmodium is diagnosed, the so-called blood smear. So let us talk more about the life cycle of these parasites and how they go about creating this horrible disease. The life cycle is shown in cartoon form here. Now, it is oversimplified for the purposes of this lecture, but I just want to hit on the main points of the developmental cycle. So it begins with the bite of a mosquito. As I mentioned, the Anopheles mosquito carries the Plasmodium parasites. The Anopheles mosquito then releases sporozoites into the bloodstream. These sporozoites travel through the bloodstream to the liver, where they then invade hepatocytes. It is here the parasites replicate and develop over the course of a little more than a week. After this period of replication, the so-called merozoites burst forth from the hepatocyte and begin a continual cycle of infecting red blood cells, the so-called asexual stage of the parasite. It is here that the parasites will invade red blood cells and consume the hemoglobin inside as a food source. This is where all the clinical manifestations of the disease occur, and this is where most drugs attempt to eradicate the parasite. Now, with some infrequent rate, by a mechanism that is frankly poorly understood, a small number of parasites will develop into a sexual form called gametocytes. It is these gametocytes that are then picked up by a new Anopheles mosquito when taking a blood meal, and the entire cycle starts again. And so, if one can treat the disease either here, or block transmission here, or block passing on the disease to new mosquitoes here, one can effectively control this disease. Now, let's take a little more close look at each stage here. So I'd like to show you a movie first. So in this movie by Vandenberg and colleagues, what you'll see is the proboscis of a mosquito, that is the needle-like, tube-like structure that the mosquito uses to suck blood of its victim, piercing the skin of a mouse. And what you'll observe are the infective form of the malaria parasites, the sporozoites, being released from the tip of the proboscis with the saliva. So let me start the movie and guide you through it. So first, what you'll see is the proboscis moving around trying to find a good blood supply. Then a second mosquito comes on the scene, and you'll see the proboscis enter here. See these small white particles that seem to be moving around? These are the sporozoites, and they exhibit what's called gliding motility, as they try and find the bloodstream to ultimately seek out the liver and invade hepatocytes. We'll let that movie run for a second so you can see the sporozoites circle around. And then finally, the proboscis of this mosquito will withdraw. Okay, so back to the life cycle. You've seen the sporozoites enter the bloodstream. From here they go to the liver, replicate, and then burst forth in a multitude of numbers to infect erythrocytes in essentially a perpetual asexual cycle. And it is here that, again, all the clinical manifestations of the disease occur. So let's focus on that blood stage asexual cycle of the parasite. This is it shown in more detail. As I mentioned before, this is the form that bursts forth from the liver, the so-called merozoites. It is these small forms of the parasite that will bind to a new erythrocyte and invade it. Now, after invasion, the parasite is called a ring stage, and that's because it resembles a small diamond ring on the inside of the erythrocyte. Then the main metabolic phase of the parasite follows. This is the so-called trophozoite phase. And after this, the parasite undergoes multiple rounds of replication to ultimately form what's called a schizont. The schizont will then mature, burst again, and merozoites are released. And this entire cycle repeats itself essentially every 48 hours in Plasmodium falciparum. So let's focus a little bit more on this stage end this stage with yet another movie. In this movie by Glashikova and colleagues, what you'll see is an infected erythrocyte, shown here, burst, spilling forth merozoites. This erythrocyte that is uninfected over here will become infected by a new merozoite that attaches to it and then enters through the membrane. So, as I start this movie, you'll see this dark spot in the middle. This is a crystal of so-called hemozoin. This is the byproduct of hemoglobin digestion that's been collected in the food vacuole of the parasite. So let's start the movie. The erythrocyte bursts, and now you can see the merozoites. Now look closely here. There goes a merozoite into the cell. Did you see that? It actually happens quite fast. Now, the real time of this movie is shown in the upper left corner. Let's see it again. The erythrocyte bursts, and watch here. There it is. The merozoite enters the blood cell. So in the matter of about less than 120 seconds, cells burst, merozoites find new homes, invade, and yet the process starts again. And as you can see, if one parasite enters a cell and many more come out, upwards of anywhere from 12 to 24, you can imagine that this exponential increase in parasites is devastating to the human host. And you can also imagine how complications like severe anemia can occur. Okay, let's go back to the life cycle. As I mentioned before, the asexual erythrocytic cell cycle of the Plasmodium parasite is where all the clinical manifestations of the disease occur. So if we are seeking to control malaria at the level of treating patients, people sick with the disease, this is the stage in which drugs must be designed to work against, to have efficacy. So let me give you a quick review of the historical nature of drugs used against malaria. Now, I'm not going to cover all the drugs, but some of the milestone drugs in malaria development. So we can begin in the 1600s with quinine. This is the most famous of all malaria drugs and comes from the bark of a particular tree found in South America. It was brought to the New World by the Jesuits, and for the last 400 years has been the mainstay of malaria treatment. Now, that being said, there are instances of quinine resistance throughout the world. Now, during the 1940s, actually during World War II, there became a need for additional malaria drugs because quinine supplies were not available to Allied forces because they came under Imperial Japanese control. Chloroquine was a German drug that was rediscovered by Walter Reed, the US Army, during an effort to find new antimalarial medications. I should mention that many of the wars throughout history have been shaped by malaria and the disease that it causes. And so there's great interest to the US Army, who puts troops in harm's way in places where malaria transmission is endemic to find new treatments. Chloroquine was one of the wonder drugs of its time. It was very cheap to make, very bioavailable, and extremely potent against Plasmodium falciparum. This was the drug of choice for a long time, but unfortunately, due to misuse and frankly overuse throughout the world, it became useless in South America and Southeast Asia, and then resistance spread worldwide, which is why in the 1970s, during the Vietnam War, Walter Reed, US Army, once again embarked on large screening programs, out of which came the new drug mefloquine, also known as Lariam. Mefloquine has been the mainstay to be used against drug-resistant parasites for some time, although now there are parasites, again spread worldwide, that have shown mefloquine resistance. This is also a drug that's not as cheap or as easy to use as chloroquine, and so is not ideal. Out of the very same screening efforts that brought us mefloquine also came halofantrin. This is a drug that was being investigated during the 1980s for use and is shown to bypass resistance to chloroquine and mefloquine resistant parasites. Yet, halofantrin also has its issues, primarily cardiotoxicity in certain individuals. And so it cannot be considered as a drug for ultimate widespread use. Now we're here in the 1990s, and the drug of choice for researchers to investigate is artemisinin. Artemisinin is a compound that was originally discovered in a Chinese herbal remedy for malaria as part of a screening program on the part of the Chinese army, and then later validated by Walter Reed in the US Army and others. Now, as you can see from this timeline, over the last 400 years, and these are not all the malaria drugs, but over the last 400 years, there have been very few compounds developed for malaria. And that's because malaria is endemic in the poorest parts of the world. And there is not a very large profit incentive for major pharmaceuticals to develop this. This is beginning to change now with the advent of greater philanthropy for malaria research and therapeutic development. Yet, I want to impress upon you that very few drugs are developed every year or even every decade for anti-malarial use. And I should also mention that the days of monotherapy are over. It is not possible for us to rely on a single drug to help eradicate this scourge. And from the lessons of HIV and other infectious diseases, we have learned that to bypass resistance or to lower the probability that resistance will occur in new parasites, we must use drugs in combinations. And so, artemisinin is being used in combination. This is a formulation called Coartem, which is a mixture of artemisinin and lumefantrine, another drug, that can be used in combination to help lower the probability that parasites will become resistant to this drug. Now, these compounds are not easy to make and they're not cheap to make, which is why there's limited availability of Coartem throughout the developing world right now. But if we are ever to hope to control malaria, we need to treat people that are sick and we need combination therapies. So a large emphasis of research in the malaria community and in my lab is to help try to develop new, cheap, bioavailable, non-toxic anti-malarial therapeutics. So the other way to control malaria, of course, is to block the route of transmission, or at least reduce the risk of transmission. So what you can see here is a mother and her child sleeping under an insecticide-treated bed net. And this is a very effective way to prevent transmission. Now, of course, the bed nets need to be maintained, and the insecticide needs to be reapplied to the bed net with a certain periodicity. Therefore, this cannot be the only method of choice for control of the vector, since leaving the responsibility of the implementation of such a public health policy to the individual is most likely not going to be successful or sustainable over the long run. Now, one of the primary weapons against the insect, the vector that transmits malaria, the Anopheles mosquito, of course, is insecticides. And the most famous insecticide of all is DDT. DDT, or dichloroDiphenyl trichloroethane, is of course one of the most infamous insecticides ever applied. And this is because used inappropriately or irresponsibly for agricultural crop spraying can result of leakage of this compound into the environment and the ecology, causing detrimental effects to many species of birds in particular. However, DDT is very cheap, very safe for humans, and can be applied indoors, inside residences and dwellings where mosquitoes might land after taking a blood meal, and used in this conservative fashion can prove to be a very effective weapon against the vector. However, because of worldwide bans against the use of DDT, there has been decreasing use of this compound. And the effects of decreasing use of DDT are shown here. So in blue are indoor residual house sprayings in South America. As you can see, as a function of time going into the 1970s here, the number of dwellings sprayed decreases. And in the red bar shows relative number of malaria cases per year. And so what you see is a very clear anti-correlation between the use of DDT and the increased prevalence of the malaria parasite in South America. And this holds true for other parts of the world as well. And so, as a result, there are new recommendations on the part of the World Health Organization. Basically, in 2006, the WHO has said effective implementation of indoor residual spraying with DDT or other recommended insecticides should be a central part of national malaria control strategies where the intervention is appropriate. And so, without the use of insecticides, we think basically that the battle against malaria will be very difficult indeed, if not totally intractable. So to summarize, controlling malaria requires certain things. We need a new generation of anti-malarial drugs that can be used in combination. Again, the days of monotherapy single drugs are over, but we need combinations of cheap, effective, non-toxic, easy-to-make drugs. This must be supplemented by an organized healthcare delivery system. The infrastructure in countries that have endemic malaria must be subsidized to be able to distribute such effective medications. If we don't treat sick people, we do not break the chain of transmission. Furthermore, we need sustainable vector control, either through insecticide-treated bed nets, indoor residual spraying, or ecological means such as draining swamps and so on. This could hopefully one day be supplemented by vaccine development. As I said before, there is no vaccine in existence today with an operational impact against Plasmodium falciparum or the other 3 plasmodia that cause malaria. Yet, we can hold out hope, hopefully, that a vaccine will be developed to help eradicate this scourge. Without these elements, the battle against malaria will continue, and that's going to be a big problem. So hopefully you've enjoyed this presentation and background about malaria, the disease, and its parasites. Thank you.